Good afternoon, everybody. I'm Jayatri, Chief Bioscientist here at the Franklin Institute, and thanks for joining me again for another conversation about the science behind the COVID-19 pandemic. It's an honor today to be joined by Dr. Paul Offit, who's a pediatrician and a vaccine scientist at the Children's Hospital of Philadelphia. And very pertinent to what we're going to be talking about today, he's one of the co-inventors of the rotavirus vaccine. So he's been through this whole process of going from basic science into the world of public health. And so we're grateful to have your expertise and insights into how this process is affecting the situation that we face today. So Paul, I wanted to start out by talking about drugs and then shifting to vaccines, because as we're seeing this parallel pipeline of just trying to develop everything that we have, both at treating the disease in people who are already infected, as well as preventing people from getting infected who have not yet not yet gotten the virus. So let's start with drugs and then think about vaccines as well, because we've heard headlines coming out about on both fronts over the past week. So with respect to drugs, the one that everybody's talking about right now is this drug called Remdesivir. We know that the mechanism by which Remdesivir works is to actually block the replication of the virus. And there were some early clinical trials that yielded some results that came out last week and garnered a lot of headlines. What do you take away from these results so far?
Right, so Remdesivir was a drug that was developed to treat Ebola virus. It didn't work. So it sort of sits on the shelf. It's a drug, it's an orphan drug. It's a drug without an FDA indication or licensure. So then the hope was that because it restricts or interferes with the enzyme, so-called RNA-dependent RNA polymerase of this virus or any RNA virus, the hope was that it would interfere with the virus's ability to reproduce itself, therefore interfere with the ability for it to cause infection. There was a press release that was issued by the National Institutes of Health about a study of a little more than 1,000 people who either were treated with this drug or not. The primary endpoint was, did it reduce the length of time during which you were sick? And the answer was yes, it did, in a statistically significant manner. Instead of being sick for 15 days, which was true for the placebo group, you were sick for 11 days, which was true of the treatment group. So that was a statistically significant difference. Unfortunately, there was no statistically significant difference in terms of death. So the antiviral did not prevent you from dying, which is really the bigger and more reasonable goal. What bothers me a little bit about this is it's sort of like science by press release. I really would like to see the data first, because the question is, did it prevent you from going to the intensive care unit? You come into the hospital, you require oxygen. Does this prevent you from going to the intensive care unit, and does it prevent you from going to the morgue? That's the question. I certainly didn't prevent you from going to the morgue. It probably didn't prevent you from going to the intensive care unit. And that's all because the way viral infections typically work is when you're first exposed to a virus, that's when you have the most viral replication. As time goes on and your immune response sort of comes up, the viral replication becomes less and less critical. So therefore, an antiviral drug isn't going to work as well later in illness. That was also true in the Chinese study, where they looked at people. It's a small study, an underpowered study, but again, people were in intensive care unit who were given Remdesivir or not given Remdesivir, the antiviral was no more likely to save your life. So I think it's a little disappointing. I wasn't terribly impressed with Remdesivir, but nonetheless, the FDA has given it, under its Emergency Use Authorization Act, the ability for anyone to use it for who's infected with this virus. But remember, it's only intravenous, so it's not going to take it home.
Right. So I want to dig into a couple of things that you mentioned there. And for anybody who's watching, please send us your questions as well. We're happy to answer them. So you talked a little bit about the course of the disease, that you at a certain point, the virus is replicating and increasing and the infection is getting worse. And then your immune system, as it kicks in, sometimes in some patients, that immune system then goes into overdrive, at which point the inflammation and all of the effects of the immune system are, maybe even more of a serious problem than the virus itself. And so is that what you mean in terms of thinking about when the drug might be effective?
No, I think the drug is most effective when viral replication is a critical part of disease pathogenesis. Remember, you want an immune response. The immune response that you make, your B cells make antibodies that attach to the virus. Your T cells, so-called cytotoxic T cells, where you sort of recognize virus-infected cells and kills them. So you want that. Sometimes as it goes along, you make an aberrant immune response, an unusual sort of immune response. And then your immune response can actually kill you, which is actually usually true. I mean, usually it's your immune response that kills you. You would probably wonder just teleologically why that would be true. Why would your immune response kill you? And I think the answer to that is your body gives you a certain period of time to rid you of this infection, whatever it is, virus, bacteria, parasite, fungus. And then if you get to the point where you're not doing very well, from the standpoint of society, you're probably better off being culled from the herd so that you don't affect other people.
From a biological point of view.
Right. I mean, it's not the kind of argument you'd make, for example, to parents in an intensive care unit, but you know, you can see where this might come from, because it really is your immune response that kills you. So then, you know, giving agents that sort of counteract part of your immune system, which has been shown for people who have this kind of overdrive, so-called acute respiratory distress syndrome, then there are monoclonal antibodies which actually can help.
And so that would be a different type of drug that you would administer to people whose immune systems are kind of gone over that edge, versus Remdesivir, which is really focusing on the viral infection itself.
Exactly.
Right. So I just want to tease apart those two different courses of action. And so we are seeing now that, you know, some of these different studies with different results. Do you think that's because they're treating patients at different points in their disease, or do we not know that because we haven't seen the data?
Right, we haven't seen the data, so you don't know. But I think it's the antivirals, monoclonal antibodies, convalescent sera are all going to work better earlier in illness, because that's when viral replication is a critical part of pathogenesis. As you move along, it's the immune response that becomes a critical part of pathogenesis, and viral replication is less important. So the earlier you give treatment, better. It'd be nice if there was an oral form of a drug that you really could give earlier. Here, you can't do that.
Right.
Intravenous, and that's only going to be administered in the hospital.
Right. So again, just to break that down a little bit, you know, we're hearing a lot of discussion about plasma from people who have recovered as well, and that's again early in the disease, where you're trying to get that immune response from people who have already recovered to maybe boost the response of people who are just in the early stages.
I'm not sure it would boost the response. I mean, the way that plasma would work, plasma from people who have survived this infection, is presumably that plasma contains high levels of antibodies directed against this virus. So again, that's going to make a difference only to the extent that viral replication is a critical part of pathogenesis. The antibodies will keep the virus from binding to cells. So cells would make virus, then the virus would be released, then it would go affect other cells.
Right.
That's where that would work. But later in the illness, when viral replication is not so important, it's not going to make that big of a difference, whether it's a monoclonal antibody, whether it's convalescent sera, whether it's antivirals. That's why the best thing is it true for all viral infections, is to try and prevent them rather than treat them.
Right. Okay, so just to kind of recap there, we've got some drugs, whether it's these antibodies from recovered patients, whether we've got drugs like Remdesivir, which are trying to keep the virus from replicating, that are all potentially effective early in the disease progression. Once you get past that point, where now this is where we're looking at the pneumonia in the lungs, where people are going on ventilators. Do we have any options of drugs that can help people at that stage?
Well, so what makes this virus weird is there seems to be a dissociation or disconnect between the level of oxygen in the bloodstream that's carried by red blood cells and the degree of lung pathology. So typically smokers, people with diabetes, people who live in high altitudes will tend to have a lower sort of oxygen saturation in their bloodstream. You know, you put that little thing on your finger, you can see the so-called pulse ox, you can see what your oxygenation is. And normally when people have typically it should be in the high 90s, high 90%, sort of 97, 98%. People who have severe pneumonia can have it like in the mid 80% range, which is really low. But here you have people who are in the mid 80% range and their lungs don't look that bad, you know, by a chest x-ray. So there's this dissociation. It's as if they've just flown at 30,000 feet without oxygen and then come and landed. And so what's going on there? And people are still trying to figure that out. I don't know what that is, but that's why people who, when they go on ventilators with low oxygen, don't do very well, because that's not the problem. Maybe they would do better in a hyperbaric oxygen chamber or something, but obviously that's limited. So right. I think about a year from now, we're going to look back on this and know what was going on, because right now it's not clear.
Yeah. So I have a question, and I think we're going to come back to this question a couple of times over the course of our conversation. What does this mean for the public? And is this drug being used to now treat people who are sick? And we know that the FDA did authorize this emergency use of remdesivir. What does that mean? Who's getting it?
I think everybody's getting it. I mean, anybody who comes into the hospital now is hospitalized either in intensive care unit or just on the floors with COVID-19 are going to be getting this antiviral. The FDA has basically allowed indiscriminate use of the drug, so that's the way it's going to be used. And I think it may prevent one being in the hospital longer, although it doesn't appear to prevent you from going into the morgue.
Okay, so that's where we are with drug development right now. Let's switch gears now to talking about vaccines. Obviously, as you mentioned, that you think that there's a lot of opportunity here that's more effective in terms of preventing the disease in the first place. So the headlines that we saw this week were that there's a group at Oxford University in the UK that has another vaccine that's gone into clinical trials in humans. And they're certainly not alone. We know that there are, many, many vaccines currently in development. And I think the last count I saw was that about 6 are in clinical trials right now. What's the biology behind how these vaccines are being developed?
Well, everything's on the table. I mean, this is a virus where you just got the strain a few months ago. You have no idea how to make a vaccine, and therefore all possibilities are being tried. I mean, there's more than 70 companies across the globe, as well as many research institutes that are looking at this. The FDA has received more than 120 investigations of new drug licenses. The good news is everybody's doing everything all at once. So should this be a whole killed virus like the polio vaccine? Should this be a live weakened form of the virus like the measles vaccine? Should it be just the protein that sits on the surface of the virus, which is that spike protein that attaches to cells, the way that we make the hepatitis B vaccine or the human papillomavirus vaccine? Should it be a so-called vectored vaccine, meaning that it's a relatively harmless virus into which you clone the gene that codes for that protein that sits on the surface of the virus? It makes it look like a crown, hence the name corona, which is the approach that's being used by Johnson & Johnson. It's the approach that's being used by the UK investigators, who have a essentially replication deficient monkey virus that they use as their sort of benign.
And so just to clarify what that means, it's a virus that's derived from monkeys, but it can't actually reproduce itself. So it's safe for humans.
Right. And then there's 2 novel approaches for which there's no commercially licensed vaccine. One is so-called messenger RNA. So you inoculate people with messenger RNA. That messenger RNA is then translated into the protein that you're interested in, the spike protein, to which you make an immune response. Or DNA vaccine, which is you inoculate with DNA that has the gene that codes for that protein that sits on the surface of the virus, that's then transcribed into messenger RNA, that's then translated into the protein. So those were all approaches that are considered everybody's doing everything all at once, because we have no idea how to make this vaccine. The second thing we don't know is what is the immunological correlate of protection. So in other words, as we know that one of the companies, Moderna, was doing the trial with the mRNA messenger RNA vaccine, that they gave 3 different doses: low dose, medium dose, high dose, looking to see what the immune response was with each of those 3 doses. And what they found, I'm sure, was that the highest dose induced the highest immune response. But what is the immune response that's correlated with protection? You don't know that. You don't know 2 critical things: how to make it and what immune response you're trying to evoke. That's why typically vaccine programs take 20 years to do, because there's so much you don't know at the beginning. Here we're trying to do it in 12 to 18 months, not knowing those 2 things. So we'll see how this plays out.
So that's an interesting point you bring up, that there are these scientific questions around the vaccine that we don't know. We don't know how to make it. We don't know what effective means. But then there are also lots of steps that that follow that in terms of actually producing and delivering this vaccine. And another headline that caught people's attention this week was this idea of this Operation Warp Speed project to try and get a vaccine ready for delivery by January. How do you accelerate this timeline compared to the traditional process by which vaccines are developed, to even think about this kind of a scale?
All right, so let's go through how you typically would make a vaccine and then what's happening here. Because if what I'm saying is correct, and I think it's fair to say I was fortunate enough to work with a team at Children's Hospital of Philadelphia that invented the rotavirus vaccine, Rotatak. That was a 26-year effort. I mean, that's not unusual for this. So what typically do you do? First, you do a so-called proof of concept study, which is to say, hopefully you have an animal model. So you take a mouse or you take a monkey, you inoculate it with this SARS-2 virus, and the animal then develops signs and symptoms similar to what humans make. Great. Now you have a way to study this virus. Then you inoculate with something that you think could be a vaccine. Then you challenge the animals with that virus, see if they get sick or they don't get sick. And then best of all, you can figure out what part of their immune system was protecting them, which you can do in experimental animals much more easily than you can do in people. Okay, now you think you have something. So then you do a dose ranging trial to make sure that you're not giving too much virus, which would be a safety issue, or too little virus, which would be an efficacy issue. You want to make sure that dose that you pick consistently and reproducibly induces the immune response that you saw in animals that was protective and that you think is going to be protective in humans, remembering you don't know that yet.
Right, but at least you have an idea of what to look for.
You have a rough idea of what to look for. Then you do phase 1 and so-called phase 1 and phase 2 studies where you do progressively larger studies of safety and immunogenicity to make sure that you don't have at least a common side effect problem and that you can consistently induce the immune response you think is going to be effective. Then you do the big phase 3 study. I mean, for our vaccine, which was the rotavirus vaccine, that was a prospective, placebo-controlled, 70,000 infant, 4-year, 11 country, $350 million trial. That's about average, okay, for a vaccine trial. Okay, so
and we've talked about randomized, controlled trials and the value of that, in these conversations before. So we can put a link in the comments to some of the conversations we've had about that and the resources we have available for people who are interested in digging into that.
So what's being done here? I think in many ways you won't see animal model studies. I mean, the UK group that has this replication defective monkey virus has done monkey trials and believes that the vaccine looked good in monkey trials. But remember, as David Weiner, who's at the Wistar Institute, famously says, he's a vaccine researcher, mice lie and monkeys exaggerate. Always do. I mean, the animal model studies are only so predictive. They give you some things to think about, but you never know till you're in people. So then you do the big phase 3 trial. This tells you that your vaccine is effective. This tells you that at least there's not an uncommon side effect problem. What's not being done here? Many of these companies aren't doing animal model studies at all. They went right to sort of small dose ranging trials, and then they just so that they can get an immune response. So now they're just going right to phase 3. I mean, it's just hold on a second. Yeah, come on in. I think there's somebody coming here who wants to meet you. There's somebody who wants to meet you. Okay, a surprise visitor, Jayatri here. Come on in. Let's see.
I have a guess.
You have a guess.
Hi, Will. So we have the second generation of Offits here. And I was lucky enough to have Will intern with me at the Franklin Institute a couple of years ago. Probably more years than it feels like these days.
Thank you so much for giving me that internship in that summer and for me.
It's always a pleasure to see you, Will. Thanks for stopping by.
Thanks, Will. So getting back to this, I think what I think this is going to play out as follows. Starting in July, I think what the National Institutes of Health is going to do is they are going to serve as an enormous pull mechanism to see how they can move this vaccine forward. I think they will test as many vaccines as companies want to be part of this trial. I think they'll share a placebo group. That makes it a little easier, I think, to move forward. Right. I think that the size of the trials that they're talking about are between 10,000 and 20,000 people. I think that in order to do that, you're going to need to have a surrogate for protection. In other words, the goal can't be to protect against hospitalization and death because you'd need, I think, a much bigger trial for that. You're going to have a surrogate for protection, which would be, for example, 10 or 14 days of cough, shortness of breath, persistent fever, something like that, to say that if you're protected against this, then you're probably going to be protected against hospitalization.
So a couple of points to pull apart here. So we did get a question asking about what phase of the clinical trials we're in now. And what we're seeing is that a lot of these are kind of combining the phase 1 and the phase 2 to do them simultaneously. We haven't yet really gotten into the large scale phase 3 trials yet. Is that correct?
That's right. That's going to start, I think, in a couple months.
Right. Okay. And it's hard to know how that how the progression of the clinical trials might predict being able to reopen society. That there's still a lot of questions that we need to answer before we can have a date to predict how the vaccine might play a role in that.
Yeah, I think it's not going to be a vaccine. I think it's going to be actually several vaccines that roll out, not only in this country, but in the world. I think in the best of world, remember, you're not going to have 330 million doses that are available on day one when they decide that this does appear to be safe and effective, hopefully. Everything has to go right in order for this to continue. I think, and one other thing before I get to who gets it, the hardest part of vaccine manufacture or development and manufacture is the making of it. I mean, mass producing vaccines is not easy. You have to have the right buffering agent, the right stabilizing agent, the right vial. You have to do real-time stability studies. You have to make sure that the vaccine doesn't degrade as you send it out there and it's being put into the arms of people, that it doesn't degrade from the tarmac to the person's arm. And you have to show that you can consistently make these lots again and again and again. I mean, these aren't small molecule products. This isn't amoxicillin where you can say 50 milligrams here and 50 milligrams here. It's the process of the product, and the process has to stay the same. So that's hard. Okay.
So just to break that down again a little bit. So you have, as we start thinking about production, there's a lot of places where, just being able to make the same product over and over again is complicated because this is making a complicated protein. The chemistry, the biology is complicated. And then you have to think about, what's involved in storage and distribution and what are conditions, because you don't want something that requires a very delicate temperature range that it has to be shipped at, because that could all go south.
Yeah, and there's so much in this to make sure you get it right. So many things have to go right. It just, I guess, is a little worrying when I hear, for example, the CEO of J&J say, we can make 1 billion doses. We're going to be out there within a year. I mean, you don't know whether your vaccine works or not yet. You don't know whether it's safe or not yet. How about a little humility? Just stand back from this process, because nature gives up its secrets slowly and often with a human price attached. So stand back, take your time, make sure it's right. Because remember, when this vaccine rolls out, I think we were talking about this before, who's going to get it? I think hopefully the CDC will define their sort of first, second, and third tier groups. The first tier group would be, for example, healthcare workers, people who work in mass transit, grocery stores, pharmacies, critical industries, let's say, meatpacking industries, and then you'll go down the list. But also, most of the people who are going to get this are going to be healthy young people. These are not people who are going to die from this virus. So you want to make sure you hold it to a high standard of safety.
So can you say a little bit more about that in terms of prioritizing, who gets that, in terms of, older people, young people, people with, people who might be more vulnerable? Who makes those decisions?
Well, I think it'll be based on the data. I mean, to what extent, for example, when these trials are done, are older people represented? To what extent are people who have certain illnesses, asthma, whatever, represented? So you can then say with at least some confidence that you can vaccinate these groups. You certainly want to vaccinate, for example, people who work in nursing homes. The question is whether or not people who are living in nursing homes also benefit from the vaccine. As a general rule, people who are over 65 years of age, with or without sort of other health problems, generally don't make as good of an immune response. I mean, things fade when you get older.
Right.
You're going to find this out, Jayatri, as you get older. Trust me on this, but things fade. And one of those things is your immune response, which gets senescent. I mean, the flu vaccine does not work as well in people over 65 as it does in people between 6 months and 18 years of age.
So as we start to think about how these vaccines might play a role in getting back to some semblance of normal, there's a lot of judgment calls that we have to make. And so, for instance, with going back to Remdesivir, I read today that the company that's making Remdesivir has released its entire supply of the drug to the federal government to then distribute along those lines. Is that typically how a drug or a vaccine would get distributed?
No, but this is an unprecedented time. So you have a pandemic. So I think here, you're not going to get, or shouldn't get, price gouging. So I mean, again, it's Gilead actually is the company that
Right.
you have to. I just think people should temper their enthusiasm about remdesivir. It's just not a wonder drug. It shocked me when it was released and the market went up 500 points. I just think that everybody's looking for the savior.
Yes.
Here. I do think you're never going to get enough population immunity to stop the spread of this virus without a vaccine. I think Bill Gates is right. The only way to really stop this virus is with a vaccine. And I'm on this working group that's put together by Francis Collins at the National Institutes of Health that includes pharmaceutical companies that are making this vaccine, the FDA, the CDC, academics. And so one of his charges initially in the first meeting was Dr. Francis Collins' charge, he's head of NIH, was that we need to make 1 billion doses. But I think that's going to be several different vaccines made by many different countries across the globe that are going to be doing this. And we may even find that there's one vaccine is synergistic with another. There's just so much unknown here that we are going to learn a lot once this vaccine already gets out there that we may not know before it gets out there. I think that's always maybe a good thing. We'll see.
So one of the things that we actually brought up in my conversation last week that I think gets to your point about how we find out the answers to some of these questions is the idea of challenge trials once you have a vaccine, how do you tell whether it's effective? Well, can you intentionally infect people with the disease and see how well they do? What do you think is the likelihood that something like that might happen in this effort to accelerate the process?
Right. Good timing. Actually, as part of this NIH group, they just formed a subgroup to look at this, and I'm actually on the subgroup, and we're meeting today at 5:30. So information for you in a few hours. But so when you challenge somebody with this virus, essentially the kind of thing you would do with mice or monkeys, right? You have now a challenge system where you can then more quickly see whether your vaccine works. It's definitely true. But other than waiting for people to get sick, which you do in the placebo-controlled group, here by inoculating people with the virus, they're going to get sick. So that definitely speeds things up. The problem with that is, and I can say that many ethicists have already weighed in on this, that they think it's an ethical thing to do. It is ethical to do this. Art Kaplan, who's at NYU Langone, has said that. Chris Feutner, I know at our hospital, Children's Hospital of Philadelphia, has said that. Because you could argue that when you do a placebo-controlled trial, you're also letting people get infected. So what's the difference? You infect them intentionally, or you let them get infected. It's sort of like the sin of omission versus the sin of commission. Somehow we view as more nefarious that we inoculate somebody with a virus as compared to letting whoever it is that makes the virus, let's say the devil, inoculating them with the virus. So, but there really is functionally no difference. The only difference is you're doing it in a controlled way. The thing that makes people a little anxious about this is there's not a rescue drug. I mean, if remdesivir doesn't protect you from dying, then know that you're inoculating people, intentionally inoculating people with a virus that is potentially fatal. Now, you're doing that to a young group of healthy people, so the odds are much more in your favor, and you can give remdesivir much earlier now that you know that the person's gotten infected and they're part of your study. So I know that there are a number of people who are pushing for this, and there will be a subgroup looking at this. I think we'll see how it plays out.
So the takeaway from all of this, it sounds like we should check our optimism about remdesivir and the impact that it's going to have, knowing that that it's not a home run. There's a lot of questions that still remain about when and how it's most effective. There is, I think, and this is where somebody asked last week, where do we find hope? And to me, I think the answer is that there are so many people who are trying to develop something, and I think the picture that you painted of the efforts to develop vaccines, at so many different points in the pipeline, all over the world, that it's not going to necessarily be one thing that solves the problem, but we're building a toolkit.
I think to try and sound a more optimistic note, see, you're a young person, so you're always like, old people are always grim. You know that, right, Jayatri? But here's what I would say. The good news is you have more than 70 companies that are interested in doing this. You have an enormous amount of money in it. You have Gates money, you have BARDA money, which is part of Health and Human Services. You have the World Health Organization starting it as a tremendous pull mechanism. So money is not the problem. Interest is not the problem. Expertise is not the problem. There's a lot of vaccine intelligence that's working toward this. And by trying to find a way, the so-called warp speed program under the administration to try and mass produce these products before you've ever shown whether they work. So that the minute you show that it works, that you can release it, which is what we did with the polio vaccine back in the 1950s. I mean, 5 companies made a polio vaccine in the 1950s without any risk. I mean, the March of Dimes said, make it. We'll find out whether it works later. And when it works and it was shown to work, then the 5 companies could release the drug. So release the vaccine. I also don't think money is going to be an issue here, because I think that these companies have received so much money from these pull mechanisms that the price is not going to be, there's no price gouging here. Plus the virus is stable. I mean, it's a single serotype virus like measles or mumps or German measles. It's not like flu, I think, where it's going to be a moving target. So that's all good news. The only
right. So just in terms of clarifying that, just means that the virus isn't changing quickly, so that right now, based on what we know, we don't have to worry that we might develop a virus or a vaccine that works, in December, but that won't be effective the following December.
I think that's right. And although it's a little obviously disconcerting that we still have no idea how to make this vaccine, we're using all possible strategies and doing them quickly.
All right. Well, that feels like a ray of hope. In an otherwise grim time. But I think we're continuing to see how the science here is advancing faster than I think anybody might have imagined under normal circumstances. I have a question here that, going back to the idea of challenge trials, as long as people are informed of the risks and rewarded or compensated for volunteering, why would anybody consider a challenge trial to be unethical?
I think most ethicists consider it ethical. I mean, and you could make the argument that when you do a placebo-controlled trial, you're asking those placebo recipients not to get a vaccine. You could argue that the challenge trial, the people are better off. One, because they're getting a vaccine, then they're being challenged, so they have have at least had a chance to be immunized by the vaccine. And if they're not immunized by the vaccine, then they've been naturally infected, and they know that in all likelihood they're now immune for the next few years. So in many ways, it's win-win, as compared to the placebo recipient who hasn't gotten the vaccine and may be challenged with the virus sort of just randomly.
Right. I mean, from what I've read, I think some of the concerns, whether or not necessarily this means it's unethical, but at least things that we should be thinking about is that, how do you inform people of the risks when there are risks that we may not know? But I think, as you said, that's true for a lot of these experimental treatments.
It's true for every experimental treatment and every vaccine.
Yeah.
When you do vaccine trials, as we did, for example, in 70,000 infants, that showed you that you didn't have an uncommon or even very uncommon side effect, but it didn't prove that it didn't have any side effect. I mean, rare side effects are only determined post-licensure. As Maurice Hilleman, who I consider to be the father of modern vaccine, famously said, quote, "I never breathe a sigh of relief until the first 3 million doses are out there."
So we've got a long ways to go. But Paul, thank you so much for joining us today, sharing both from your past experience as somebody in this field, as well as your involvement with all of the efforts that are going on right now to tackle this crisis. Really appreciate your insight.
My pleasure.
Thanks a lot, Paul. Talk to you later.
Wait, don't go away. So can I still talk to you?
Yes, of course.
Okay,
and I almost forgot to mention one thing to our audience, which is that we're in these really unprecedented times when I feel like we're saying that phrase over and over again. But Paul, your conversation today has really brought to the forefront that science is more important than ever. That no matter all of the other challenges that we face, ultimately, we still have to figure out the science. And that's the goal of the programs that we're doing here. Our weekly COVID-19 updates are an essential part of helping our audience understand the process that we're involved in. while many of our other Franklin at Home programs are here to help you discover the curiosity as you're stuck in your own house. And so tomorrow, there's actually a larger philanthropic effort called Giving Tuesday Now that is designed to help nonprofits like the Franklin Institute who are doing the best we can to stay engaged with our communities and make that difference. And if you're considering a way to give back for the work that the Franklin Institute is bringing to your home, we'd be honored if you'd consider a donation today or any day. So thank you for considering that as part of Giving Tuesday Now. Hopefully we'll see you here next Monday. Thanks for joining us, and we'll close our conversation now. But keep sending us your questions. Thank you.
So can you and I talk now, or
We're just gonna wait for Katie to end the live stream.
I'll wait for the live stream to shut down.
It's fun to see Will.
So when can I talk to you? Can I talk to you all? Can I talk to you without everybody hearing it? Does that ever happen, or what? Yes.